What's going on, guys? I'm going to make a video about MCAT mnemonics. I have a lot of mnemonics and I realized that MCAT is very memorization heavy. If you could find mnemonics, then it's going to be easier for you to kind of have that information in your head instead of just rote memorizing everything, because that's going to be super difficult. And I pretty much have mnemonics for every single subject, so I got some for biochem, for all the sections, right? So since it's a lot, I'm going to break it down to subjects, and within subjects I may even break it down to different parts. This is biochemistry part 1, mnemonics. So let's get started. rRNA for eukaryotes. Let's say that eukaryotes is 2, so it's 2, 4, 6, 8. So the rRNA is 20S, or is 2, so it's 4S or 40S, 60S, 80S. For prokaryotes is 1 because it is smaller than eukaryotes, which is 2. So for prokaryote it is 1, 3, 5, 7. 30S, 50S, 70S. Eukaryotes it is 2, 4, 6, 8. Prokaryotes it is 1, 3, 5, 7. Next one, for purines, it is pure as gold. So purines are adenine and guanine, and then cut the pie. Pyrimidines are cut the pie. So it is cytosine, uracil, and thymine. Then bit of orgo, which is also like in the biochemistry lane. More bonds to oxygen means that there's oxidation. Less bonds to hydrogen, it is also oxidation. And vice versa, more bonds to hydrogen is reduction, and less bonds to oxygen is also reduction. It may not be super accurate organic chemistry wise, but it is good enough to know for MCAT. That'll help a lot of mental energy for you. Next, the Na and K ATPase, so that's just sodium, potassium, ATPase. How do you know what goes out and what goes in, right? So the way I think of it, sodium is bad for you because that raises your BP. So you want sodium to go out. Potassium is good for you, or it's in bananas. You want potassium to be inside you, so it's going to go in. Sodium goes out, potassium comes in. And just remember 3, 2, or 3 over 2. So 3 Na out, 2 potassium in. For DNA replication, as you should already know, 2 strands, right? DNA polymerase is going to read up, 3 to 5, and it's going to read up, and then it's going to write down 5 to 3. That's how you know which way it's going. Norepinephrine and epinephrine, they both end in, right? Norepinephrine and epinephrine in. So it's inside, so it's adrenal medulla, because medulla is on the inside, the adrenal glands, right? So inside. Then for amino acids, know the name BAPAN, B-A-P-A-N. I will link a video by Shomu's biology, and he's going to explain how to memorize the amino acids. So he uses the mnemonic BAPAN, and I think he could better explain it than I can. Next, for the short codon, August. AUG is the short codon. So August, school starts in August. So short codon, AUG, August. Then ADH, always digging holes, because the ADH is going to retain water. So if there's holes, then water is going to zip in there, right? More proteins, MKD, I think of it as my kinky dance, myosin, kinesin, and dynein. Ligase, they're going to seal molecules together, so it's ligature, ligase, right? Lig. Then GABI, G-A-B-I. Glucagon is alpha cells in the pancreas. Insulin is the beta cells of the pancreas. Next one is Leo is a jerk. So Leo, that is L-E-O, loss of electrons is oxidation. A is anode. So now you know that loss of electrons is oxidation, and that happens in the anode. Jerk, G-E-R-C, gain of electrons is reduction. Jerk, there's a C at the end, that's in the cathode. So Leo, loss of electrons, oxidation in anode. Jerk, G-E-R-C, gain of electrons, reduction at the cathode. Next one, ATGC. A bonds with T, thymine. G bonds with C. And obviously for RNA, T is going to be replaced by EU. And then remember these numbers, and I'll explain what these numbers mean. So 1 1 1 1 1 2 2 1. So A is 1 1, T is 1 1, G is 1 2, C is 2 1. ATGC 1 1 1 1 1 2 2 1. So what does it mean? It's either acceptor or donator. A is 1 1. So the first one in the 1 1 is a acceptor, because A comes before D. Adenine is going to accept 1 hydrogen bond. And then it's got another one, right? So adenine is also going to donate 1 hydrogen bond. So how do you accept? You accept if you have either an N or an O in your molecule. That's how you accept the hydrogen bond. How do you donate? If you have an H in your molecule, then you could donate to do the hydrogen bond. So adenine is going to have 1 acceptor, 1 donator. So it's going to have either 1 N or O and 1 H. for adenine. So T is also going to have 1, 1. So 1, 1, 1, 1, 1, 2, 2, 1. So T is going to have 1 acceptor, 1 donator. So 1 acceptor is going to be either N or O, and 1 donate is going to have an H. 1, 1, 1, 1, 1, 2, 2, 1. So now G is 1, 2, right? So it's going to have 1 acceptor and 2 donators. So G, ATGC, 1, 1, 1, 1, 1, 2. So 1 acceptor, so that is N or O, and then 2 donators, so 2 H's. Now C, 1, 1, 1, 1, 1, 2, 2, 1. So C is 2, 1. So it's going to have 2 acceptors and 1 donator. So it's going to have 2 either N or O and 1 donator. So that's going to be 1 H donating. If you just memorize that, ATGC 1, 1, 1, 1, 1, 2, 2, 1, then you know which molecule is going to have what. Next, how do you know whether purines or pyrimidines have 1 or 2 rings? The way I think of it is pure as gold. Purines, gold. I say that gold is heavier. If it's heavier, it's got more rings, which means it's got 2 rings. So purine, pure as gold, so adenine and guanine, they'll have 2 rings. And then the rest is 1 ring. The pyrimidines, it'll just have 1 ring. So now that you know that purines, pure as gold, have 2 rings, and then you could combine that with what I said earlier about ATGC 1, 1, 1, 1, 1, 2, 2, 1, then you know exactly which ring is what by looking at how many N's or O or H's they have and how many rings they have. Let me know if that's confusing. I'm trying to make it as simple as I can. Next, for the Krebs cycle, please officer, can I keep selling soap for money, officer? You could do that. I know that a lot of people also say, please officer, can I keep selling sex for money, officer? The reason why I'm saying please officer and not officer in the beginning, it goes from pyruvate to acetyl-CoA. So please officer, right? Can I keep selling soap or sex, whatever makes it easier for you? Can I keep selling soap for money, officer? The N, the O is oxaloacetate. Next, for snowdrop, that's the mnemonic, and that is for the southern blot, northern blot, western blot. You have all these blotting techniques, right? Which one is for what? So just know snowdrop. S is southern, D in the drop is DNA. Then N in the snow is next, right? So that is northern, and then you have R in the drop, that is RNA. Then you have snow, then the third one is W, right? Western blot, and then in the drop, P is the last one, that is protein. So DNA, RNA, protein, snow. Snow drop, southern, northern, western, DNA, RNA, protein. Follows the central dogma as well. DNA to RNA to protein. Next, prolactin is the production of milk. So P, prolactin, production of milk. And then oxytocin, I think about it as oozing, oxytocin, oozing. So it's gonna ooze out, it's gonna have the milk let down. Prolactin just produces the milk. Oxytocin is actually gonna cause the milk to ooze out, right? So it's gonna have the milk let down. Next, for this one, don't really have a mnemonic for this one, just kind of like know it. Parathyroid hormone is going to enhance osteoclast activity, and calcitonin from the thyroid gland is going to do the opposite. It's going to inhibit the osteoclast activity. Next, it's kind of coupled, right? Osteoclast, class C, collapse. So it's going to collapse the bones to release calcium into the bloodstream. So PTH, parathyroid hormone, is going to enhance osteoclast activity, which in effect causes bone collapse, so the calcium goes into the blood from the bones because the bones are collapsing. Osteoblast in your bones, B, blast, is build. So osteoblast is build. So it's going to cause the calcium to go from the blood to your bones to build the bone. Next, flat peg. Just know flat peg. Flat is the first word. That is your FSH, LH, ACTH, TSH. Okay, flat. Peg, it is prolactin, endorphins, and GH, growth hormone. FSH, LH, ACTH, TSH, flat. Peg, prolactin, endorphins, and growth hormone, GH. Flat, right, all those hormones are in the anterior pituitary gland. And PEG, that is the posterior pituitary gland. Flat are all tropic hormones. What that means is that it's indirect. So the hormones from anterior pituitary gland is going to first activate some other gland, and then the effect is going to be from that secondary gland. So all of the flat hormones are not direct. They do not cause the effect directly. However, for PEG hormone, which is from the posterior pituitary gland, they're direct. Those hormones cause the effect directly. I'll give you an example. TSH, thyroid stimulating gland. TSH goes from your anterior pituitary gland to your thyroids, and then the thyroid gland produces your thyroid hormone. The TSH does not create thyroid by itself. It's not a direct cause and effect. Right? It is indirect. That is a tropic hormone. That's all the flat hormones. But for PEG hormones, it is direct. Prolactin. That causes production of milk directly. Next, for antibodies, know the term degam or degam, D-E-G-A-M. DEG, deg, that is one type of antibody, one similar structure. Then A, it looks different. M also looks different. So DEG is the smallest one, and then A is bigger than deg, and then M is the biggest one. DEG-A-M, degam. Then B cells versus T cells, where they're maturing. B matures in the bone marrow, B and B, and T cells, they mature in the thymus, T and T. Just want to make one clarification. Both B and T cells are made in the bone marrow, but they mature at different sites. Next, for some immunology, eosinophils. They are for parasites. They kind of like, attack parasitic organisms. The way I know that eosinophils is for parasites is EPA. I get a lot of emails from EPA. That's the Environmental Protection Agency, EPA. So I think that E is eosinophils, P is for the parasites, and A is attack. All right, next, DAKE, D-A-K-E. I think about it as Drake. So DAKE, D-A-K-E, and that is all your fat-soluble vitamins. Vitamin D, A-K-E, fat-soluble vitamins. So DAKE, Drake. Your water-soluble is B and C. So the way I remember is Batter soluble, right? Batter, water, and soluble. S and C, they sound similar. So Batter soluble, B and C. Glycolysis, you should definitely memorize all the enzymes for glycolysis. So I think a lot of the prep companies and in general, a lot of the advice is that you could just get by just knowing the key enzymes, right? But I think it is just easier to know every single enzyme in glycolysis, because a lot of the times you'll have questions that require you to go into the passage and look for the enzyme that causes some effect, right? So if you already know it, then you don't even have to go to the passage. It's almost like pseudo-discrete question. So I would rather over prep in content than having to go back to the passage looking for the answer. I would highly recommend just know every single enzyme in glycolysis. And the way you do it, or the way I did it, was I had this memorized. So GGFF, F, and then G, 13-BPG, 3P, 2P, P, P. On the other side, DHAP, DHAP, and then glycerol. So if you just memorize that and then just keep practicing, just literally just like take out a notebook and just start writing all the steps of glycolysis, I guarantee you within 2 hours you have memorized. And you want to do this exercise, if not every day, at least every other day. You want to memorize the glycolysis completely, and also the Krebs cycle, because it's going to help you a lot. It's just like one less thing to worry about during test day, so you don't have to go back to the passage to look for the answer. It's a pseudo-discrete. Then nucleoside, S side. So I think about it as second, 2. Nucleoside, second, 2, S. So it's going to have a sugar and a nitrogenous base, 2. Nucleotide, T is for 3. You're going to have the sugar, nitrogenous base, and a phosphate group. There's 3 groups, right? So that is nucleotide, 3. Nucleoside, 2. Alrighty, let's go to the digestive system. G cells, gastin. GG, it's pretty easy. Chief cells is pepsin. The way I think about it is the chief thing to drink is Pepsi. Chief cell, pepsin. Histone, heterochromatin. H is for hard, so it is hard to transcribe. So if it is a heterochromatin, then it is hard. Euchromatin, it is easy. E for easy, so it's going to be easier to transcribe. And estrogen is going to establish the wall. So estrogen, EST, is going to establish EST, the wall. Progesterone is going to protect the wall. Progesterone, PRO, pro, is going to protect, PRO, the wall. Next, I was getting confused between centromere and centrosome a lot. But if you think about it, right, in a chromosome, centromere is in the chromosome, okay, because kinetochore is a protein that's also in centrosome. So centromere and kinetochore, they have the ear ending. Centromere, kinetochore. Just to clarify one thing, centromere, it is part of the DNA. So centromere is DNA. Heterochromatin, it is in the DNA. Kinetochore, that is a protein. And then centrosome is on the sides. So the way I think about it too is zome, S, zome, sides, S. Okay, centrosome on the sides, centromere, kinetochore in the middle. Next, ADHV. So ADH is also called vasopressin. So ADHV is kind of like play of words with ADHD. ADH hormone is also called vasopressin, ADHV. Next, synthase is no energy. No energy is required for that enzyme, and synthetase does require energy. So if you notice, synthase is smaller than synthetase. So if synthetase is larger, it's going to require energy. So synthetase requires energy. Synthase does not require energy. Next, when you look at a brain, it is gray. The inside part, which you're not seeing, that is white. So the inside is white brain, the outside is gray brain. How do you know which axons are myelinated? The way I think of it, right? The inside is your old brain. So that acts way faster than the outside brain, which is your newer brain. All the logic and everything, cognitive thinking that humans have, is all new to the brain. So it is on the outside. All the emotion stuff and all the survival stuff that we have from the past is inside the brain. That is the white brain. When something happens, right, immediately you act on your emotions. That is faster. So that is derived from the inside, the white brain. That means that the axons that are inside is faster, and so it's going to be myelinated. So white brain inside is myelinated axons. Gray brain outside, it is unmyelinated axons. T3 and T4 are from the thyroid and they regulate the temperature, T temperature. PMAT and MCAT. So PMAT, that's a mnemonic to know all the cycles or all the steps of mitosis. So prophase, metaphase, anaphase, telophase. Prosthetic group, which is a cofactor, right, is extremely tight, if you know that. And the way I think about it is that if you have a prosthetic on, right, let's say you lost your limb, then if you have a prosthetic leg on you, it's really tight, right? It's not going to be loose, right? So a prosthetic group is tight, and that's a cofactor. It's going to be a tight cofactor. All right. Holo enzyme. Holo sounds like whole. So once the enzyme is whole, it's got its cofactor and whatnot. It is whole. It is fully equipped now. So holo is whole. Whole is ready. And the apo enzyme is just the opposite. It does not have the cofactor and it is not ready to do its enzymatic function. CCAT, C-E-K-A-T. CCAT, that is the 5 structural proteins. And then stop codons, it is U go away, right? UGA, and then I say Y. And because you are annoying, UAA, and you are gross, UAG. And that is the end of part 1. So we'll do another one.